Remember this visual from about 1 or 2 lessons ago? Well, we're going to get into what this visual is. We're going to talk about all this vocabulary, and we're going to start learning how to make a Punnett square. This scientific tool that doesn't look scientific, it looks like a bunch of boxes, but it is used so that we can trace genetic traits from parent to offspring. So we're going to use this, except I need to get rid of this board. This is much better. Okay, so let's talk about, let me just show you an example of what a Punnett square does. Have you ever thought about the odds of being a boy or a girl? Well, those odds can be explained pretty easily by using a Punnett square. So let me go ahead and create my boxes. Little lopsided, I don't like that. Let's change that. That's better. Okay, so here's my Punnett square. What am I going to fill up here? Well, I need to put the genes, the pair of genes, or an allele up there. So we're going to put the female's genes, female, up top. And I always like, in my class, I just say, let's always put the female's genes on top because it's easier for me to correct. You don't have to. Other places don't do that, but in this case, we're going to do that. So the female's genes are going to go on top, which means the male's genes are going to go on the side there. And what genes are we going to talk about? Well, let's talk about sex genes, the genes that make a female, and a male. If you are a female, you have XX. I put an allele here, and now we have a pair of genes. See, throwing in that vocabulary that you need. On the side, male's genes can be also an X, but there's also the Y inside this, or inside males. So females are, now let's talk about some vocab. They are the same type of genes, homozygous. Males, different, heterozygous. All the vocab coming into play right now. Now let's cross these genes and see what we get. So if we take the genes from the female and the genes from the male, homozygous. And these genes just happen to be dominant. So homozygous, dominant. Let's cross again. Homozygous, dominant. We now have 2 chances to possibly have a female. Let's cross one more time here and one more time here. Heterozygous, dominant. So let's talk about our genotypes. What 2 genotypes do we have? Well, our genotype is either XXXX or XY. What about our phenotype, the physical characteristics that we can see? Well, if we have XX, that is going to be a female. If we have XY, that's going to be a male. What is the percentage? What's the chance of having a female? Well, 2 out of 4 together would be 50%. Which leaves, there's always 100% in everything, so which leaves the other 50% to be male. Okay, so we used all of our vocab. We've got homozygous, heterozygous, capital letters are dominant, smaller letters are recessive, genotypes, the types of genes inside here, phenotypes, the physical characteristics. Each one of these genes is an allele. So all that vocab coming back, and now we're going to do some practice with this right now so that you get better at Punnett squares and that you're able to do them on your own. So I'm here to work with you doing it says more Punnett square practice, even though we haven't even practiced the first time. But this was one of the best examples I have for us to walk through and to learn how to create a Punnett square, find the genotypes, the phenotypes, and the percentages, and use all our vocabulary at the same time. So here's what I'm gonna do. We're gonna talk about the first example problem here, and we're gonna talk about the vocabulary and see if you know the vocabulary. In this example, it says that rabbits with black fur is dominant over rabbits with white fur. So if we're gonna write, and in this case, they said if you cross a male, capital B, capital B, capital B, capital B is the genotype, and we're gonna put the male, and normally I would have you put the female up top and the male on the side because that's just the way I've been grading during the years I've been teaching this. But in this case, they don't do that. They put the male across the top. So we're gonna follow what they did. They put capital B across there. Those are the genes for the male dominant black rabbit. So what do we call this? Capital B, capital B. It has a name. Did you say homozygous dominant or dominant homozygous? You can say it either way. If you did, that was correct. Down the other side, we've got the female's genes, and it says that she is capital B, small b. Capital B, small b. What are we going to call capital B, small b? They're different, but there is a dominant allele. So that name is, I hope you said heterozygous dominant. Dominant heterozygous. Okay. Genes for the male, genes for the female. We can tell by these two dominant genes, the male is black. We can tell by the dominant gene right here that the female is black. Let's cross these genes now. When we cross them, we get what you see in the boxes. And now we can talk about the different offspring that is possible from rabbits who have homozygous dominant, heterozygous dominant. Let's write down our genotypes. So what do we have? We've got a capital B, capital B, capital B, small b. We've got two different genotypes here. Capital B, capital B, capital B, small b. Genotype, the types of genes that are possible from this cross. What are the phenotypes now? Well, what are we going to call this? The same and dominant should be a homozygous dominant, and that would mean black fur for that rabbit. Down here, capital B, small b. That's right, heterozygous dominant. Also, we have one dominant gene, two dominant genes. Dominant genes push out recessive genes, and then we end up with the dominant trait, which is black fur, coming through. Percentage of the chance that we will have black fur, that's pretty easy right now. 100% chance if we have a homozygous dominant, heterozygous dominant pair of rabbits that have offspring will have black fur. Okay, welcome back to the Punnett Square Practice Hour. What we're going to do and talk about right now are cabbage butterflies. That's right, cabbage butterflies. Cabbage butterflies have one of two colors. We've got white wings or we've got yellow wings. White wings happen to be dominant over yellow wings. So in cabbage butterflies, white wings are dominant to yellow wings. If we have a capital W, small w, cabbage butterfly, what do we call capital W, small w? Dominant heterozygous. And we cross it with a small w, small w. What do we call these two together. This is a new one for us. Yep. Recessive, homozygous, homozygous, recessive. So I can tell right now that up here, this butterfly is going to have white wings. Why is that? There's a dominant gene. This butterfly doesn't have the dominant gene, which means that butterfly carries recessive genes, and this butterfly is going to have yellow wings. So let's cross these together and find out what possible offspring can have here. So when we cross capital W, small w, and it is good form for us as a class to write the capital letter first if there is one. It'll help us all out. It'll help me out when I'm looking at your papers. Small w, capital W, small w. All right, we've got a couple different genotypes happening here. So let's take a look at what we have. How many different genotypes do you see? You should have said 2. Good for you if you did. We're going to write them down. There's our first one, capital W, small w. What are we going to call this? That is going to be dominant heterozygous. I heard you out there shouting it. How about right here? We've got another. What are we going to call that one? That's right, homozygous recessive. Okay. Phenotypes, the physical characteristics. Well, we've got a dominant gene right there. That dominant gene is going to push out the recessiveness, and we're going to get the dominant trait, which happens to be white wings. This one, however, there's no dominant. Both recessive genes show up. And when two recessive genes show up, the recessive trait comes through, and that's when we're going to get yellow wings. Last part, what percentage of the offspring are going to have white wings or yellow wings? Well, we only have a box of 4. This whole box equals 100%, which means each block's going to be 25%. So we're going to have capital W, small w, happens twice, 2 out of 4, or 50%. There's other 2 boxes are small w, homozygous recessive, 50% there as well. The next example we have for us to do together, and then I'm going to let you do some parts of this on your own, and then finally, eventually, a couple of these completely by yourself and see if you are getting the hang of Punnett squares. Okay, so in dogs, there is a heredity type of deafness. And that hereditary type of deafness is caused by a recessive gene. If the dog ends up with both recessive genes, the dog can possibly be deaf. So we have 2 dogs that are going to carry the recessive gene. And those 2 dogs are going to be written, come on, capital D, capital D, small d. What are we going to call capital D, small d? It should be dominant heterozygous. They both have a dominant gene, and then they have an allele that is different from one another. Capital or dominant allele, recessive allele, together they are heterozygous. Both dogs carry a dominant and recessive gene. Now, let's do our cross. All right, you can tell by our box here, we've got a multitude of different genotypes. So let's go through and put our genotypes down, and as we put them down, we're going to name them. Our first genotype that we come across is capital D, capital D. Capital D, capital D. What are we going to call capital D, capital D? You should have said dominant homozygous. They are the same. They are both dominant genes. Homozygous, dominant. The next one I see, got these 2, capital D, small d. Capital D, small d. How do we name this one? That is right. It is dominant heterozygous. Dominant gene, these two genes, or these two alleles are different, making it heterozygous. And then finally, we've got everything going on in this one. We've got small d, small d. What would you name that? It should have been homozygous recessive. Recessive homozygous. Now, what are the phenotypes going to be? It says that the recessive gene causes the deafness. So, in the case of having a dominant gene, you're going to get a dog that can hear. So we have homozygous dominant. That is a hearing dog. Heterozygous dominant. That is a hearing dog. And then the two recessive genes do show up in this offspring. So that offspring possibly will be deaf. So let's take a look at our percentages now. We've got 3 different genotypes, 2 different phenotypes. What are the percentages of having a hearing dog? Well, I've got 1, 2, 3 out of my 4 are have dominant genes in them. So these 2, we're going to be with 75%, and the possibility of having offspring that is deaf will be 25%. Now, let me put it this way. If a female pup did have 4 pups, female dog did have 4 pups, it is possible that this math doesn't work out this way. It's possible for all 4 dogs to be capital D, capital D. It's possible, not likely, but possible that all 4 are recessive. So just because we do the math this way, this is in a perfect world. And as you know, things happen in our worlds. Okay, let's talk about a pet I've never had, my kids have never had. We're going to talk about guinea pigs. So in guinea pigs, apparently short-haired guinea pigs, or short hair in guinea pigs, is dominant over long hair in guinea pigs. So we've got short-haired guinea pigs, we've got long-haired guinea pigs, and now we're going to do some crossing to see what happens when we have a capital S, capital S. Go ahead, you can name that one. You should know what it is by now. Homozygous, dominant, dominant homozygous. So we're going to cross a dominant homozygous with, a recessive homozygous. Okay, so go ahead. I want you to do this cross by yourself. All right, this is the cross you should have come up with. Ended up with capital S, small s in every single box. What is capital S, small s? Go ahead and name that for me. That is right. It is dominant heterozygous, and it happens in all 4 of our offspring. Capital S, small s. It's always good to be careful when you're writing these letters to make a distinction between capital and small. Over exaggerated if you need to. And that's it. That's all we have. We've got 1 genotype, which means we're going to have 1 phenotype, and that phenotype is going to be capital S, small s, dominant. Short hair is dominant. And that happens in every box, so it's going to be 100%. We've talked about tongue rolling in the past and whether someone can roll their tongue or not. Yes, my daughter can and I can't, and you saw that in a video before. However, I told you my son is not a tongue roller. So how is it possible to have a tongue rolling child like my son, or a non-tongue roller like my son and me, with a tongue roller like my daughter? Well, real quick, and this is not in here, I wanted to show you what that example looks like. It looks like this. This is their mom. That's me. When you cross those, that's what you get. And my son apparently inherited this right here, which makes him a non-tongue roller. So it is quite possible for people to have traits that is different from their family, just depending on the genes that get passed on. So in this case right here, we are talking about tongue rolling. Suppose a man who is capital T, small t for tongue rolling, marries a woman who's capital T, small t for tongue rolling. Well, let's go ahead, and I've told you we're gonna put the male's genes on the side, the female's genes at the top. I want you to do that and then make the cross. Okay, did you make that cross? If you did and it looks like this, you are understanding Punnett squares and you're well on your way to acing this stuff. Okay, so we've got a bunch of different genotypes and we've got some phenotypes there. I would like you to use your information with tongue rolling being dominant, non-tongue rolling being recessive. I would like you to fill out the next part by yourself. And this is what you should have written down. We've got two types of dominant genes. We've got dominant homozygous, dominant heterozygous, both tongue rollers. Together, 3 out of 4 of those offspring possibly have the tongue rolling gene, and that is 75%. And we have 25% as being a non-tongue roller. So we're back at it again with the guinea pigs. And I did not know that there were rough coats and smooth coats for these things. So apparently rough coats, like this one, which is all swirly and cowlicks, like it's popping up all over the place, that is dominant over smooth coats, like this fellow right here. But I've never seen one of these rough-coated guinea pigs before, so that was strange to me that gene, that rough-coated one, was dominant. But you're flying solo on this one. I want you to go through from start to finish, fill in the boxes, get the genotype, phenotypes, percentages, and let's see how you do on your own. Okay, from start to finish, I hope this is what you ended up with. We had our homozygous dominant, heterozygous dominant, breed. They passed on their genes to their offspring. We ended up with 2 different genotypes, homozygous dominant, heterozygous dominant. Both of those genotypes are rough phenotypes, rough fur, and when's that gonna happen? 100% of the time. So I hope this beginning process of Punnett squares, I hope this helps you. We've got a couple assignments coming up that you're gonna do on your own. We're gonna take a little stop down to Bikini Bottom and do some Bikini Bottom genetics with SpongeBob, Patrick, and all the other ones that I can't remember. So this is due. I would like you to submit this just to show me that you did the work with me today, and then I'll give you your points for it.